The second part of the central dogma is translation, where RNA makes proteins. Proteins are large complex molecules. They do most of the work in the cells and are required for the structure and function, regulation of the body tissues and organs. The building block of proteins are amino acids. It means amino acids combine to make proteins. 3 codons called a triplet codes for one particular amino acid. For example, there are 900 bases. There are 9, if you count this A, C, U, G, if there are 900 bases and you want to make a triplet, there will be 300 codons. And those 300 codons will be translated into 300 amino acids. For example, if you take the triplet AUG, which is on the messenger RNA, it codes for the amino acid methionine. Now go to the next triplet CAA on the messenger RNA. It codes for the amino acid glutamine. The next triplet CCA on the messenger RNA codes for the amino acid proline. C on the messenger RNA codes for the amino acid histidine. AAG on the messenger RNA codes for the amino acid lysine. Now, you know that the messenger RNA was formed in the nucleus and it left the nucleus through the nuclear pore and it has entered the cytoplasm. So you have to imagine that this is the cytoplasm. Okay? Now, once the messenger RNA leaves the nucleus through the nuclear pore and has entered the cytoplasm, it moves about in the cytoplasm till it meets the ribosome. Ribosome is a protein manufacturing unit which contains the ribosomal RNA. Once it meets the ribosome, translation begins. Now, a small subunit of the ribosome, this is a small subunit of the ribosome, attaches itself to the messenger RNA. And it keeps moving on this strand until it meets the start codon, which is AUG. Now, this movement on the messenger RNA is known as translocation. Now, once it meets the start codon AUG, the larger subunit of the ribosome and the transfer RNA join it. Any tRNA cannot join it, as each amino acid has its own tRNA molecule with the anticodon for that amino acid. What does that mean? Now, for the AUG codon on the messenger RNA, which codes for the amino acid methionine, UAC anticodon of the tRNA binds to it. The moment it binds to it, it drops this amino acid, breaks off and leaves to pick up another amino acid. Now coming to the next codon, CAA on the messenger RNA, which codes for the amino acid glutamine. GUU anticodon of the transfer RNA binds to it. Once it is bound, it drops the amino acid glutamine, breaks off, and leaves to pick another amino acid. And this process continues, and the amino acids proline, histidine, lysine, yes, are dropped. Now, once they are dropped, you have the RNA, that is the ribosomal RNA, which will help them to form peptide bonds. Now, these peptide bonds will result in a chain. Like for example, I've shown you over here, there is a chain. Here there is a chain. See the amino acid methionine, glutamine, proline, histidine, lysine, glutamine, serine. Now they are all connected by a bond which is supplied to them by the ribosomal RNA. So now they have formed a chain, and this is called a polypeptide. Now it can contain at least hundreds of amino acids. Now this process will not go on and on and on. It will stop. It will, amino acid will be formed here, amino acid will be formed here, amino acid will be formed here, here, here. Now when it comes to this, it means stop codon. Now the stop codons can be UAA, UAG, and UGA. Any one of this is a stop codon. Now when it comes to the stop codon, there is no tRNA that recognizes this. So what happens? The ribosome knows that translation is complete, and it will release the polypeptide which is formed. It will release this polypeptide which is formed. Yes, and it will move to the next messenger RNA which has to be translated. The polypeptide chain which is formed may undergo additional processes. It may get folded due to interaction among the amino acids. It may bind to other polypeptides or different types of molecules such as lipids, which are fats, carbohydrates. Many of them will travel to the Golgi complex, which is inside the cytoplasm. And what does the Golgi complex do? It packs them into membrane-bound vesicles before they are sent to their destination to perform their work.